Hello Year 10, this is just a little video to help you complete the command words task, which is our follow-up task from our face-to-face day last week. The first two questions are really straightforward. So the first one is name the structure in plant cells where chlorophyll is found. So that's just going to be simple fact recall. Use your revision guide to help you. Use the notes we put together in the face-to-face to help you with that one. Next, we've got to explain the function of chlorophyll. So remember that word function just means job. And then it's for 2 marks. So we're obviously going to have to explain that in a little bit more detail. So make sure you're fully sort of talking about what you know about chlorophyll and its role, its job in photosynthesis. Those two should be straightforward. The next question, it says a student wants to study the effect of carbon dioxide concentration on the rate of photosynthesis. Okay, so we know carbon dioxide is going to be one of those things that affects rate of photosynthesis because it's one of the reactants. So if we have less of the reactants, we're going to be able to make less of the products, which are glucose and oxygen. And remember that word rate of photosynthesis means how quickly photosynthesis is occurring. And we usually measure that by counting numbers of bubbles of oxygen produced, or we can measure the volume of the gas produced. So a student is wanting to investigate that, and they've been given all of these different bits of equipment. What you need to do is choose which equipment would be useful for you doing this investigation. Now, to answer this question, I want you to think back to the required practical. We watched the video on the required practical in the face-to-face session, but most of you have done the practical as well. I've linked in a copy of the video of the required practical into the Show My Homework that links to this lesson as well, so you can review that practical. Now, when we did the practical, we were looking at the effect of changing light intensity on the rate of photosynthesis. Remember, we had a light source and we put some pondweed into some sodium hydrogen carbonate solution, and we basically moved that pondweed different distances away from the light source, and we counted numbers of bubbles of oxygen produced per minute. In this question, we can see that this student was studying the effect of carbon dioxide concentration. So it's slightly different. What they're going to do is they've got now a different independent variable. Okay, so the independent variable, remember that's the one that you change in the investigation. The independent variable is not light or distance from the light source, as was the one where we investigated. This time it's going to be sodium hydrogen carbonate concentration. So what you need to do is think about, right, okay, so how could you vary the concentration of that? And if that's your independent variable, your dependent variable is going to be the same. It's going to be number of bubbles of oxygen, because that's how we measure rate of photosynthesis. You've got to think, what else have you got to keep the same? So think about all of the other factors that might affect rate of photosynthesis. And if we didn't control them, they might affect our results. So to answer this question, you've then got to think, right, what apparatus would I need to do that? This picture might give you a bit of a clue. There's a big list there, and you've got to pick out all the ones that would be useful. Now, there are 5 marks there, which suggests that you probably need to pick out at least 5, well, 5 of them. That's what you're going to get the marks for. And then for the next question, it asks you to give a reason for each piece of equipment you chose. So for that bit there, all you really need to do is just say why you're using that bit of equipment. Okay, so if you think about the reason for using a piece of pondweed, which I'm sure everybody picked out from the list, obviously the pondweed is going to be the thing that does the photosynthesizing, and so it's going to produce the oxygen bubbles that you're going to count to measure your rate of photosynthesis. So you try and pick out a reason for each of the ones that you chose. Next question is quite straightforward, and here it says define. Now we know that a limiting factor's got a dead set definition. So go back, look in your notes and see what your definition of a limiting factor is. We know when we talked about limiting factors, we talked about 4 things that would affect the rate of photosynthesis, either increasing it or decreasing it. It says other than carbon dioxide. So we've got 3 others to choose from. There's only 2 marks available for this question, so you need to pick out any 2 of them. So that one should be straightforward. Next question, working down. Here we can see, for question 7, it says the graph shows the effect of increasing carbon dioxide concentration on the rate of photosynthesis. So here's our increasing CO2 concentration, and here's our rate of photosynthesis. Now remember, this graph's got that typical shape that we've seen before in other graphs when we're looking at limiting factors and rates. What we've got to remember is, in this part here, where we're seeing an increase in rate as concentration increases, okay, and then we're seeing in this part here, no matter how much more you increase the concentration, the rate doesn't increase any further. Now, don't forget, when you're talking and explaining this graph, it's really important to explain what is the limiting factor between here and here, i.e. what's the limiting factor where you can see on the graph the rate of photosynthesis increasing, and what factors could be limiting in this space here. Now, I'll give you a clue. Remember, we talked about this in the session, where the line is increasing and shows an increasing rate, it's always this factor that's labelled on the x-axis that's limiting. Between this point here and this point here, where the rate is staying the same, this factor, and it doesn't matter what it is, in this case it's CO2, is no longer limiting. And we know it's no longer limiting because even though we're adding more of it in, it doesn't make any difference to the rate, it's stopped going up. So another factor, another one of our ones that we would have mentioned here, must be limiting. And so you say which ones are limiting here. So fingers crossed that should help you have a go at explaining that shape of the curve. Then on to the other side, we've got a bit of an extension question for question 8. But have a go at it, give it a go. It says the combustion of fossil fuels raises atmospheric carbon dioxide levels. We know when we burn any fuel, it produces CO2. Evaluate how this might impact on food production around the world and use your knowledge of photosynthesis. So try to remember that the food products that we consume are often plant material. So think about what we know plants do to carbon dioxide levels and see if you can explain it. I don't want to explain that one too much more. I'll help you that when I go through the answer. Next one is again another bit of an extension one, but again worth having a go at. It says marram grass grows on sand dunes. Marram grass has stomata that are sunken in pits and not on the surface of the leaf. Explain how these sunken stomata help the marram grass to survive in windy, dry conditions. So this one here is a little bit of a kind of link to other questions that we did and work we did earlier on in year 10 when we looked at that process of transpiration. So remember, stomata are those tiny holes in the undersurface of the leaf where water can escape through. Now, if you sink your stomata deep into the undersurface of the leaf, so not right on the surface, you're going to reduce the rate of diffusion of water coming out the leaf, and so you're going to reduce your rate of transpiration. So that's a pretty tricky one. You can have a go at writing down an answer for that one. And then this final one, everybody should be able to have a go at again. It says respiration occurs in all living things. Compare the processes of respiration and photosynthesis. So for this one here, there are 2 big things I want you to talk about. I want you to talk about which one's endothermic and which one's exothermic. And then I want you to talk about how their reactants and products compare. Now, I know we haven't done respiration yet, but you will remember what the word equation was for that. And you'll have some ideas for that one. So have a go at that one there. But again, that's pretty tricky. Okay, so pause the video now, have a go at them, and I will go through the answers. Okay then guys, let's go through the answers. So first ones are really easy. Name the structure in the plant cells where chlorophyll is found. So we know that's in the chloroplast, 1 mark. Explain the function of chlorophyll. So we've got 2 marks here, so we've got to write in quite a lot of detail. So it absorbs the light and the light provides the energy for the reaction, okay, for that 2nd mark. Next one, we needed to identify the equipment that we would need to use in this reaction. So remember, in this investigation, you're asked to study the effect of carbon dioxide concentration on rate of photosynthesis. So the first thing we're going to need is we're going to definitely need a piece of pondweed. we're going to need something to change the concentration of the carbon dioxide. And remember, to do that, we use this chemical sodium hydrogen carbonate. Okay, so we've got the pondweed, we've got the sodium hydrogen carbonate, which we can change in concentration in this tube here. We need a boiling tube to put both of them in. And remember, as this photosynthesizes, little bubbles of oxygen are going to be produced, and we can either count them directly, or we can measure the volume of water displaced, or the volume of sodium hydrogen carbonate displaced, or we could use a funnel to collect this gas. So that would be another mechanism. Other things we're going to need is, because we're only changing this in our investigation, we're only changing the concentration of carbon dioxide, we need to keep all the other factors that would affect photosynthesis exactly the same. So we need to control the temperature. So we do that by sticking our whole investigation into a big water bath. And we would add into there water at 25 degrees C, and we would make sure that we put a thermometer in it. And if that temperature went down in our reaction, we'd top it up with a bit more warm water. So we maintain that water bath at 25 degrees. That will control and keep the temperature the same. Another big variable we need to keep the same is the light source. So we'd use the same lamp, and we'd position that lamp a set distance using our meter ruler. We'd set it a set distance away from our pondweed, and we keep it that distance every time we did the investigation, with every concentration of sodium hydrogen carbonate and when we did all our repeats. Okay, the other things you need to keep your same in your investigation would be the amount of chlorophyll available. Well, we're going to do that by using the same size piece of pondweed, or the same piece of pondweed, in every bit of our investigation. So that's the kind of setup that we have. So if we look at our answer, you should have ticked or selected some of these bits that are on here. Basically, in terms of mark scheme, you were able to get 1 mark for each bit that you correctly selected. So that would get you 5 marks there. And then if we look at the next page, do a quick turn over. On the next page, you need to then, let me see all of this in one shot. You then need to say why we need each bit of equipment. So what I recommend you do, although I've talked through it on the other side, pause the video a second and read through each point. You can see after each point I've put a mark, and you can go through and check your marks. Okay, so pause the video when you're ready. So now on to question 5, we've got to define the term limiting factor for photosynthesis. So remember, that's a factor that when it's in short supply restricts the rate of photosynthesis. Now, I know when we did it in our booklet, you might have copied it straight away, and you might out your booklet, and you might have said a limiting factor is one that stops the rate of photosynthesis going any faster. That would be absolutely fine for that one as well. Any of those definitions would be fine. Next one, 2 factors other than carbon dioxide that affect the rate of photosynthesis. So dead easy: temperature, amount of chlorophyll, light intensity. Doesn't matter which 2 you picked, any of them would be fine. Now the questions start to get a bit trickier here. For this one here, we've got to explain the shape of the curve. So it's not just about describing it, although often you do get some credit for describing it. It is about explaining why what you're describing is actually occurring. So the first thing we can say for the first stage between here and here is that as concentration of carbon dioxide increases, the rate of photosynthesis increases, but it only happens up to a point. Now, obviously, the reason that as this increases is that you're providing more carbon dioxide for the reaction. But what we can see is at higher concentrations, at concentrations higher than 0.1, the rate starts to level off because CO2 is no longer the limiting factor. So CO2 is limiting between here and here, but it's not between here and here. So what we get for the third marking point is to say another factor is now limiting, and we can state an example of those factors. So maybe actually now the temperature is limiting, or the availability of light is limiting the reaction and stopping it going any quicker. We know it's not CO2 because we're adding an extra of that, and it's not actually making it go any quicker. So there must be another factor now. Now, what we could do to see if it was that factor is we could perhaps raise the temperature by 10 degrees. And if we raise the temperature by 10 degrees, we might see that actually, yeah, the rate of photosynthesis goes up. Okay, so hopefully that one makes sense. On to the next question. This was a really hard one at the top. You had to sort of think, and we will do lots more on this in year 11 when we talk about the impact of having lots of CO2 in our atmosphere. But this question was asking you about, if you've got more CO2 around, how might it impact on food production? So the other thing to remember is that it's an evaluate question. So it means that you have to put both sides of the argument. So you need to say how it will positively affect food production in the world, and also what might be the negative or the downside of that. So what we would recognise is that if we've got more carbon dioxide, we can carry out more photosynthesis. Now, the more a plant photosynthesizes, then the more they can grow. They're going to make more glucose. And remember, we know if they make more glucose, they can make more cellulose for their cell walls. They can make more lipids or fats and store them. They can make more starch and store it. They can make more amino acids by combining the glucose with the nitrates to make those amino acids and build them into proteins. And overall, that just gives us a load more growth. So overall, we get more crops grown. So they're your kind of 3 marks for the plus side in our evaluation. The downsides of it is that we know that if we've got a higher CO2 level, that might cause us some pollution. And CO2 is one of our acidic gases, which causes acid rain, which can damage plants directly. Now, I wouldn't have expected you necessarily to have known this. So if you did get any of those marks, very, very well done. Okay. Next question was asking you about how sunken stomata might help marram grass to survive in windy, dry conditions. Well, remember, when we sink in our stomata, that means that you trap a little moist area just inside the stomata, and as a result, you reduce the water loss from a plant. And obviously, if a plant if it's not losing water, it's not going to wilt, and it's still going to be able to carry out most efficient photosynthesis. So that was a bit of a link to the module that we did right at the start of the year 10, looking at the structure of the leaf and the importance of those stomata. Next one we're on to is comparing, or the final one, respiration occurs in all living cells. Compare respiration and photosynthesis. So, reminder, respiration and photosynthesis, we can compare them initially on whether they're exothermic or endothermic. So remember, respiration's exothermic, so energy is released to the surroundings in the form of heat, really. And photosynthesis is endothermic, it's taking in energy in the form of light from the surroundings. Other thing to remember is that if we look at the word equations, we can do a direct comparison. So you'll recognize this is the word equation for photosynthesis. This is the word equation for respiration. We can see that in photosynthesis, our reactants, the things we need for the reaction, are carbon dioxide and water. Whereas in respiration, in aerobic respiration, we produce carbon dioxide and water. We can see in photosynthesis, glucose and oxygen are produced, whereas in respiration, this one here, glucose and oxygen are required in aerobic respiration. So either of those comparisons will be fine. Now, when we've done a bit more work on respiration, you might be able to pick out a few more comparison points. So that's it for this video. Don't forget, if you're doing the extension task, crack on with that. But hopefully you've managed to work through this and you understand what you've done. Okay, well done.